Okay, ladies and gentlemen, welcome. So in today's video, we are going to look at the concept of DNA isolation. So DNA is genetics material that is actually transmitted from parent to their generation or across their generation. And very importantly, DNA, it has so many applications. You can use DNA to determine paternity. You can use DNA to determine maternity. You can use DNA in disease diagnosis. You can use DNA to treat a disease. You can use DNA in personalized medicine. You can use DNA in organ or tissue transplanting. So DNA, it has so many applications like in cloning and gene expression and forensic. So it has so many applications. So in this video, we are going to look at how to get DNA out of the cell. That is DNA isolation or DNA purification. So scientists generally um, involved in molecular biology techniques where they actually isolated DNA. So as we said, actually this DNA is usually reside in the nucleus of a cell, and it of course, this DNA, it is found in the nucleus of the cell. Like, for example, here you can see that this is the nucleus of the cell. And of course, so for you to get the DNA, you need to open up the cell, open up the nucleus, and then you open up the chromosomes, which subsequently where you are going to actually get your DNA. So the DNA is packed in the chromosome, and then the chromosome packed in the nucleus, and then the nucleus packed in a cell. So for we to get the DNA, we need to open up the cells and get the DNA alone, and that is actually DNA isolation or purification. That is to get the DNA in a pure form, that is in highly purified, in highly purified form. So for you to do that, you need to open up or you need to remove all other component of the cell, and it is only the DNA that you are required. So in this actually video, that is what we are going to look at. And we have different sources of DNA. You can get DNA from different sources. You can get DNA from plant. Even from plant, you can get DNA from different species of plant. You can get them from flowering plant, from non-flowering plant. So you can isolate DNA. You can get DNA from there. So one of the source of the DNA, that is where you can get DNA. You can get it from plant of any species, because DNA is the language of life. So it is present in every living organism. So you can get DNA depending on what you are interested on. Then, of course, you can get DNA in animals. You can get DNA in animals like human. So you can get DNA from humans, and you can get DNA from any other animals like chimpanzee, sheep, cats, and so many other organisms. You can get DNA from them. You can also get DNA from bacteria. So therefore, we have different sources where DNA can be isolated. So you can get it from plant, from from human, and then from bacterial cell. Although when you are trying to isolate DNA from human, it's not the same techniques or it's not the same method that you are going to apply in regards to isolating DNA from bacterial cell, because for you to open up the bacterial cell, you need some uh, extra enzymes or buffer before you break down the cell wall. And of course, in humans, we doesn't have cell wall. So that is why isolating DNA from human is much more easier than isolating DNA from bacteria and plant, because plants, they are highly rich in cell wall. So these are actually some of the major sources of the DNA. So we usually isolate DNA from human, we get it from bacteria. You can even get DNA from virus, because we have DNA virus. So we can get DNA from virus. So these are actually the sources of the DNA. And in addition, then what are the sources of DNA in humans? So you can get DNA from human hair. You can get DNA from the saliva. You can get DNA from sperm. You can get DNA from urine, and then you can also get DNA from blood. So this is actually, so therefore you can get DNA from the following sources in humans, that is from sperm, from urine, blood, hair, saliva. You can all get actually the DNA from all these cells. So these are the sources of cells for DNA extraction in human. So then in addition, what are the importance of the DNA extraction? So of course, the DNA isolation allows for the analysis of genetics material. So if you isolate DNA, you can be able to carry out analysis of some genetics material. For example, in disease diagnosis, you can actually isolate DNA, identify a particular genes that is related to a particular disease. Like for example, in humans, we have a disease called sickle cell anemia. So you can isolate the DNA from human, target hemoglobin gene, and then you now send the gene for sequencing. After you sequence it, then from there you can see whether there is a mutation in the gene or not. So that is one of the major application of DNA extraction. You can extract DNA to analyze your genetics material for diseases or for treatment and so many other things. And then in addition, the the DNA extractions, it has vital applications such as cloning, sequencing, and gene expression studies. So the DNA, you can isolate DNA for you to carry out genetic cloning, or you can sequence the DNA, that is to know the nucleotides that are present in your specific target gene or the expressions. So that is if you want to see how a particular gene express. Like for example, melanin in human is a gene that is associated with skin color. So if you want to know how much of the melanin you produce in your body, and that is what you call gene expression at a specific time. So you want to know how much of the melanin gene express or produce at a particular period in time. That is what you call gene expression and other studies. So then accurate DNA extraction can in fact reliability or reliability of experimental result. So when you are trying to extract your DNA, extracting your DNA, if you actually follow the protocols accurately, there is a likelihood that you are going to have highly pure and high yield of your DNA. And when you have problem with your DNA purity, and you also have problem with the concentration of the DNA you extracted, it usually affects your downstream application. Downstream application, it can be sequencing, it can be PCR, it can be gel electrophoresis. So if you actually do your DNA extraction accurately, so there's a likelihood that you are going to have reliable results, reliable experimental result. So this is also another application of the DNA extraction. We can use it in monitoring response to disease. Like for example, if you actually identify new drugs, so if you give that drugs to a particular patient, you can use it to check the therapeutic response of that particular drugs. And in addition, we have prenatal screening or prenatal genetic screening testing. So if you have a child, if you have, of course, in a pregnant woman, you can actually get the DNA from the child, from the baby in the stomach, or you can actually get the DNA from the pregnancy of the woman to identify the genetic or risk factors or genetic risk factors, like for example, cardiovascular disease. You can actually get the aliquot of the DNA from the baby, from the pregnant woman. So after you get the DNA, you can screen whether the DNA of the child is actually having a mutation in some genes, like for example, in hemoglobin. So you can check as part of the prenatal screening to check the baby in the pregnant woman whether the baby is actually having a chances or it's actually at risk of cardiovascular disease, at risk of sickle cell disease. So you can do that. So this is actually one of the application of the DNA extraction. It also helps in screening for therapeutic targets. That is, if you want to develop new drugs, so you can actually isolate DNA for target. And at the same time, screening, detection, and early diagnosis of disease, you can also use DNA. You can also monitor disease recurrence, like for example, prevalence. Like for example, if you have any particular disease and you don't know how to monitor that disease, then you can isolate the DNA from the infected individual, and then from there you can detect whether the person is actually infected with a particular disease or not. So this is actually some of the applications of DNA extraction. So what are the steps in DNA extraction? Starting with the phenol-chloroform extraction method. So there are actually four basic steps in DNA extraction. So the first thing is when you have your cell, you need to lyse. That is, you need to break down your cell. So how do you lyse your cell? You actually lyse your cells through different methods. You can actually lyse your cells using physical method, or you can lyse it using enzymes. You can lyse it using lysis buffer. So after you lysis, or that is after you open up the cell, then the next thing you incubate the cellular debris. So after you incubate, so after the incubation, then the next thing, the cell lysates, of course, after you incubate it, and then, first you have your cell. So after you have your cell, then you add SDS, that is sodium decyl sulfate, and then proteinase key. So the sodium decyl sulfate is actually a chemicals agent. It is actually detergent that is used to break down the cell membrane of a cell. And then proteinase is an enzyme that degrades proteins in the cell. So after you have that, then you now incubate the lysed cell. So after incubation at 65 degrees centigrade, usually at 65 or around 65 degrees centigrade, so you incubate it at this. So after incubating them for some time, you also add phenol-chloroform, and then you vortex, that is, you shake, and then you centrifuge. So after you do that, the proteins, lipids, and other compounds will all be sedimented. They will all precipitated. They will go down. And then the DNA will be left as the sediment after the centrifugation. Then the sediment will be transferred to another Fendok tubes. After you do that, then you now return the aqueous phase of the DNA by rehydrating. Because after you actually transfer your DNA, then you can add buffer precipitate down your DNA. After it precipitates your DNA, then you remove the supernatant. And then you now add rehydration buffer, where you are going to rehydrate your DNA. So let's actually look at the steps more closely. So you can see that when we said cell lysis, it means to open up the cell, that is to break down the cell membrane. So when you will break down the cell membrane, of course, the content of that cell will be released into the tubes that you are trying to isolate the DNA in. So of course, the DNA is present in the nucleus, and of course, the nucleus contains chromosome, and then the chromosomes contain so many components like telomere, centromere, and of course, there is nucleosomes, there are histone proteins, and all these, they are all together with your DNA. So if you want to isolate your DNA, all these components, you need to remove them. You need to remove all of them. So that is why now, let's see how we can actually lysis, that is, open up the cell. So the first steps to break down the cell membrane or to break down the cell is you can use physical method. Like, for example, you can use high pressure. A high pressure can be applied to break down the cell membrane, or freeze and thawing. Or you can use ultrasonic homogenizers. You can use pestle and mortar. You can use blender, and you can use also agitation. So all these are the physical methods that are used to break down the cell. You can use high pressure, freeze and thawing, ultrasonic homogenizer, pestle and mortar, blender, and agitation. So you all need these physical methods to break down the cell. So after them, then you can use enzymes. Enzymes can also use to break down the cell. And one of these enzymes, we have lysozymes and lyticase enzyme. So lysozymes and lyticase enzymes, they are enzymes that are required to break down the cell wall in bacteria, fungi, and yeasts. So you see, lysozyme and lyticase, they are enzymes that require to remove the cell wall in bacteria, fungi, and yeasts. Then apart from that, there is also cellulase, which is also another enzyme. So cellulase is also an enzyme that require to break down the cell wall in plants. The cell wall in plants. And then apart from cellulose, then the next one is chitinase. So chitinase is also another enzyme that use to break down the cell wall, but in fungal organism, that is in fungi. And then we also have proteinase K. So the importance of proteinase K is to actually degrade all the proteins that are associated with your DNA. So when you open up or when you break down the cell membrane, so the content will be released, and that content contains protein and non-protein component. So to remove the protein component of that DNA or of that lysis, you need proteinase K. So proteinase K will degrade the proteins, including histone and other non-histone proteins. So it will break down all the proteins that bind to your DNA, and this ensure that the DNA is released and free from other proteins. So that is why you use proteinase K. So once you use the proteinase K, the histone protein and other non-histone protein, they will all be utilized to break down the proteins and make the DNA free. So then the next thing is lysis buffer. So you can also use lysis buffer to break open the cells, because this lysis buffer contains so many components. It contains detergent. So the major detergents that are used majorly in the DNA or in breaking down the DNA, we have number one, we have what you call SDS. So SDS is called sodium dodecyl sulfate. So you can use sodium dodecyl sulfate, or you can use CTAB. CTAB means cetyl trimethyl ammonia. SDS is sodium dodecyl sulfate, and then CTAB is cetyl trimethyl ammonium. So this is actually some of the detergents that usually use to break down the cell. And then the next thing after, because these are the component of this lysis buffer. So lysis buffer comprises detergent, chelating agent, and buffer. So the importance of the chelating agent in this lysis buffer is to chelate magnesium ion. So they will bind to all the available magnesium ion. So once they bind to them, then the DNase, which is the enzymes that require to digest DNA, will not longer bind to magnesium because there is no any available DNA. Sorry, there is no any available magnesium ion. So therefore, the DNA polymerase, the DNase, that is deoxyribonuclease enzyme, cannot find the available magnesium to bind. So if there is no available magnesium ion, it means that the DNase will not be active. So it will remain inactive. Therefore, this will prevent the DNA from degradation. So, and then the next one is buffer. So in the lysis buffer, we also have buffer. So this buffer is important in stabilizing or in actually maintaining the pH value and also stabilize the DNA. So this is actually the major, these are the major steps that are used in cellular lysis, that is breaking down the cell. We have physical methods. You can use enzymes. You can use lysis buffer. And of course, we also have ribonuclease, which I actually didn't mention. It is important. So remember, actually, this gentlemen, when you are trying to isolate DNA, sometimes there are also proteins together. So you use this RNase enzyme to digest the RNA. So usually when you are trying to isolate protein, when you are trying to isolate DNA. So the DNA and RNA are always together. They are always together. So how do you remove the RNA so that you left with the DNA? That is when you are going to add the ribonuclease enzyme. Ribonuclease enzyme. So after this ribonuclease enzymes, then of course the RNAs will be degraded and be removed. So then that is actually for the lysis method. So the next thing is, of course, on incubation. Incubation. So after you lysis, or after you add all these components that are actually involved in breaking down the cell, so the next thing is actually to incubate. So you take the cell lysate. When we say take cell lysate, it means that after you lyse the cell completely, then you incubate the lysate at a temperature like 65 degrees centigrade. So this temperature also is very important that will help the further break down the cell and release the DNA. And how this incubation achieved. So it's achieved by, number one, this incubation will aid in the digesting the protein components in the lysates. It will also aid in suspending lipids and also break down the cellular material. So that's the importance of this incubation. So all the proteins will be digested, and then in addition, the lipid will be suspended. So like for example, look at it here. Let me just give this example here. You can see clearly after, from here, you can see after you now incubate. After this, you incubate. So then the next thing after this incubation, ladies and gentlemen, you can see clearly, okay, so the incubation is usually carried out in in incubator. So this is incubator. So you can take your DNA in the tubes and then you set this incubator at 65 degrees centigrade, and then you add, okay, sorry, and then you add your DNA here. So you can actually allow it to, you can incubate it for 65, at a temperature of 65 degrees centigrade for 1 hour or for 30 hours, depending on the types of, or depending on the source of the DNA. The incubation is also depend on the source of the DNA. Is it from plant? Is it from animal? Or is it from fungi? Then after that, then, so this is the importance of the incubation, digesting proteins, suspending lipid, and then you will break down cellular compound. So the next thing is actually is to add phenol-chloroform and then you centrifuge. So the importance of adding phenol-chloroform is to remove proteins and lipids from the DNA solution. So that's why you add phenol-chloroform and then you centrifuge. So when you add phenol-chloroform, this is what you are going to observe. Look at it. All the proteins and the lipid will settle down, and then you will now left with DNA. So after they settle down, and then you now have the DNA as your sediment, then the next thing is to precipitate the DNA. So after you add phenol-chloroform and then you centrifuge, the proteins and the DNA will go down, and then you now elute, or you now transfer your DNA supernatant to another tube. So after you do that, then the next thing, you also add ethanol. You add ethanol so that you precipitate the DNA. So the DNA will settle down. The DNA will settle down. So after the DNA set down, then the next thing is to add is to add another ethanol. And the major importance of adding this ethanol is to remove salt and other impurities. And then lastly, after the salt and other impurities are removed, then the next thing is to rehydrate the DNA, like freeze EDTA. So now, because the ethanol is to wash the salts and other impurities that are still attached to the DNA. So then the next thing is the dehydration. So after you now wash, then you add another trace EDTA buffer. So the importance of adding this trace EDTA buffer is to rehydrate the salt again. Sorry, the DNA again. So after you did that, then what is the next thing? Is also the other downstream application. Other downstream application, which can be PCR, it can be gel electrophoresis. That is actually the down application. So you already rehydrate your DNA using this trace EDTA, that is TE buffer. So after you do that, then the next thing, you now store it at -20. That is at a temperature that is a freezer of -20 degrees centigrade. Then you store it there. So after you store it, then you now allow, or you now actually leave it there until when you are going to carry out other further analysis. And if you are not actually going to do any further analysis, although before then, after you successfully identify the DNA, you can also measure the concentration as well as the purity of the DNA using nanodrop spectrophotometry. But at this stage, we are not going to do that. So we can use nanodrop spectrophotometer to measure or to assess the quality of your DNA based on the concentration as well as the purity of the DNA. So ladies and gentlemen, this is actually based on phenol-chloroform. So you see, let me just briefly come back to this. So you see, after you have your cell, you need to add SDS and proteins or any other enzymes, because this is the step where you are going to open up your DNA. And I mentioned the different ways that you can actually lyse or break down your DNA. You can use enzymes, you can use physical method, you can use like Festal and Mortar and a physical method. You can also use Lysis buffer. So you see, after you lyse, then the lysate will be incubated at 65 degrees centigrade. Then after you do that, then you now add phenol-chloroform for text, and then you centrifuge. When you said for text, it's like shaking. Centrifuge is actually a machine. A centrifuge machine is a machine that you use to sediment or to separate two different solutions. The heavier one will settle down, while the those that are less heavier will be at the top. So that is why here, after adding phenol-chloroform, vortexin, and centrifuge, then the DNA and proteins are all settled down. So we don't need them. So, and then the next thing we now elute. We now take the DNA into another fresh tube. So after you do that, then you now wash your DNA with ethanol, and then you will suspend the DNA with trace EDTA buffer. So this is actually briefly on this, ladies and gentlemen. So, but it's not all. There are also other methods that you can extract DNA, and that one is usually used by kits. We have what we call DNA extraction kit. So the DNA extraction kit is a kit that used with the manufacturer instruction. There is a template usually when you isolate the DNA. Sorry, when you buy the extraction kits. So it's usually using a kit. So when you get your DNA isolation kits, then there are protocols that they will tell you, okay, follow this step, do this, do this, do this. And if you do that, at the end, you are going to successfully have your DNA. So this is actually a typical examples of that method of DNA isolation using DNA extraction kits. So you see here is after the tests, that is, you have your sample here. Let's say this is a sperm cell. Then you now complete it with about 400 microliter with extraction buffer. So you see you have your sperm cell, for example, and then you now add 400 microliter. And then after you add the 400 microliter of the extraction buffer, then the next thing is to add 20 microliter for K. And the importance of that is to break down the cell membrane and the proteins that are needed, that are not needed. Then after you do that, then the next thing is incubation. So you need to incubate it, of course, at 56 degrees centigrade for 10 minutes. So see this one, this incubation here is 56 degrees centigrade. And then the one that I actually set from the previous slide is 65 degrees centigrade. So it can be 65, it can be more than even 65, it can also be less than that, but depend on the types of the tissue that you are using to isolate your DNA. Then after that, then you add ethanol, then the ethanol will precipitate it. Then after the precipitating, then you now actually centrifuge at 6,000 times X. Then after you do that, then you now add wash buffer W1 and W2, and then we elute our DNA. And then later on, you see at this stage is the rehydration. So the rehydration is by adding 50 microliter of buffer AE. So this is actually briefly on how to isolate DNA, ladies and gentlemen. I hope you'll find this video very interesting. Thank you and see you next in my video. So don't forget to subscribe and join